plug in 12 and we don't have heating. So now you can see that we really have a cold spell here. So thank you very much for agreeing to speak. And next, hour and a half is yours. Thank you so very much. Please go ahead.
Thank you, Vinod, very much. And thanks everybody for the invitation to participate in this chronobiology school. Can you hear me okay outside?
Yeah.
Great. So thanks everybody for staying so long. I know you've had a big day there at the conference. The good news is that the days are getting longer and it should warm up soon. What I was tasked with is talking to you today about the suprachiasmatic nucleus. So in the next 45 minutes or so, I'll try and review a little bit of history and talk a little bit about current research. So the hero for the next hour is going to be this little tiny structure in the brain, which when you zoom in and label with fun techniques like rainbow, you can see the individual cells that are going to be the heroes for the next hour. So I'm going to dedicate this talk to two people. Steve Brown, who you heard about at the beginning of this conference, I think, and Dr. Michael Manafort. Both of these folks passed away this year and were extraordinary people in my life as a developing scientist. Mike Manafort was there when I was a postdoc and really imparted on me many important lessons on how to be a good scientist. And he's sort of my scientific grandfather. I'm sorry, it keeps changing slides. I'm not touching anything. And then Steve Brown was sort of my scientific brother and died tragically at a young age. And one of the things I want to impart on the students in the class is that these schools really become incubators for the future of the field. This is a photo from a chronobiology school that was held in Berlin. One of the things that was amazing about Steve and Mike was their dedication to teaching the next generation of scientists, and especially to students from all over the world, male and female. They really saw the future of the field as everybody is welcome. And they made me feel welcome, and they made me want to participate in schools like this. So I welcome your questions, and I look forward to interacting with you after the course is over. I also bring you greetings from Nikhil Mukesh, who I met at an Indian Chrono School, and he is now a postdoctoral fellow in my lab, doing machine learning analysis of daily rhythms of behavior and the neural circuits that underlie those daily rhythms. So here we go. This is what I want to talk about today. Evidence that the SCN is a central circadian pacemaker. Real-time reporters study circadian clock. Evidence that the SCN is made of many oscillators. Evidence that neuropeptides perform the role as circadian pacemakers in the SCN. And then I'll talk a little bit about the future and how I and the future scientists in the field are thinking about the challenges ahead. So you have learned a lot in this course, I imagine, about circadian rhythms in the cellular, molecular, and hopefully structural basis of rhythms in a variety of organisms. I'm going to be focusing in on the central circadian pacemaker in vertebrates today that drives daily rhythms in things like body temperature and hormones, and locomotor activity and sleep-wake, and how those depend upon how those rhythms can persist even in constant conditions to anticipate the challenges of the day. So for me, I have just experienced the surge of cortisol as I woke up this morning, and you guys are now experiencing the relatively soon rise in melatonin as you prepare for nighttime. So back in the 1950s, folks were trying to understand where did these rhythms come from. And it was sort of a radical idea that it might come from a particular place in the brain. So researchers like Victor were doing experiments where they were performing lesions into different parts of the brain and trying to abolish things like daily rhythms and locomotion. Victor never found the pacemaker, although he found, for example, that the eyes were required for daily rhythms to synchronize to the light-dark cycle. So the eyes are required in mammals for synchrony, and that's a little bit different than all other vertebrates, and in fact, many other species on this planet which have photoreceptors all over their bodies. Mammals, it seems, have a light pathway from the eyes to the brain that's really important. And so that knowledge inspired folks to do what we call following his skinnogram. So Arnold Eskin was a researcher in the field who drew this really simple diagram where he said there are inputs, pacemakers, and outputs from the pacemakers. And if we could follow the inputs in or the outputs back, we might be able to find the pacemaker. So folks reasoned that they could follow the retinal input pathway from the eyes to the brain and find the central circadian pacemaker. And so the eyes project into the brain and they make connections through the optic chiasm down here at the bottom of the brain. And then make connections into structures in the hypothalamus, including here, the suprachiasmatic nucleus. So the SCN is this tiny little structure about millimeter by millimeter by millimeter in diameter. And researchers in 1972 developed methods to lesion the SCN. And what they did was they lowered a wire into the brain and they heated it up. And that hot wire destroyed the cells located right on top of the optic chiasm. So Irv Zucker's lab and at the same time Bob Moore's lab published independently that when you lesion the SCN completely, you abolish daily rhythms in things like sleep, wake, and cortisol secretion. So this suggested that the SCN is really important for these rhythms. But does it tell us that the SCN is really the pacemaker? Or could it be that the SCN is downstream of the pacemaker and preventing the pacemaker from communicating with things like daily rhythms and sleep and hormones? So researchers like Bill Schwartz, and Mike Menaker shown here at another conference, seated together, were really pioneers in the next step in demonstrating that the SCN really is a pacemaker. So for something to be a pacemaker, it needs to fulfill a couple of different criteria. One, it needs to be required for rhythm. Second, it needs to be rhythmic in its own behavior. And third, it needs to determine the period of the behavior. And so here we go. Bill Schwartz was interested in the very early stages of functional brain imaging. And as a young researcher at the National Institutes of Health, he was working with researchers who were developing a technique to image the brain called 2-deoxyglucose. In 2-deoxyglucose imaging, you could inject radioactive glucose into the brain in a form where it can't be metabolized. So it's taken out by metabolically active cells, but then it stays within the cells. And then if you kill that animal and use the tissues as a way to develop film, you actually put the tissue right onto film. The radioactivity exposes the film, and you end up with an image of where the metabolic activity was high. And what Bill between Mike Menaker and one of his graduate students at the time, Martin Brown. They discovered a mutant hamster, and that mutant hamster you may know about is called the tau mutant hamster. It had a heritable mutation in its genome that caused it to have a free-running period as a homozygote that was about 20 hours instead of the near 24-hour rhythm of the wild-type hamsters. And the stories behind this are really fun to share with students. Martin brought this mutant hamster to Mike Menaker and said, "This is really interesting." And Mike said, "Oh, you should breed it and see if it's heritable." Years later, another student came to Mike Menaker and said, "I found this hamster with a 20-hour period." And Mike said, "What do you want to do?" And they said, "I want a new hamster. My hamster is broken." And it really sort of highlighted the importance of how you need to see opportunities when they present themselves. When things are not as you predict, you should study them further. So they did, and in fact they took advantage of this mutation later in a transplant experiment. Here, what they did was they repeated the SCN lesion experiment that you saw earlier, now on hamsters, that destroyed daily rhythms in locomotion in the hamsters. But then they transplanted in an SCN at this point to the hamster. And what you see is a gradual locomotor rhythm. And these are two examples, one hamster and another hamster. You can see the rhythms are restored in both cases. But what's striking is the rhythm in this case is restored with a short period, and the rhythm in this case is restored with a long period. And the reason behind this is that they swapped the SCN. They took the mutant SCN and transplanted it into the wild-type hamster. But when they take a wild-type hamster and transplant in a wild-type hamster and transplant in a mutant SCN, the period is determined by the donor SCN. The SCN carried with it all of the machinery to determine the behavior of the hamster. So now I want to take you many years into the future and talk about how people began to explore the role of different cell types within the SCN. So the SCN is actually comprised of about 20,000 or so neurons on the left, 20,000 or so neurons on the right side of the brain. In addition, there are about 1,000 astrocytes and several other cell types in small numbers. For many years, the field is focused on the idea that the SCN has some organization to it. It's organized into what people have described as the shell and the core based upon things like neuropeptide expression. So across many species of vertebrates, we find vasopressinergic cells on the top and VIP, or very important peptide, vasoactive intestinal polypeptide expressed in the ventrum. So down here is where you would find the optic chiasm. The nose is coming out towards you, the tail is headed into the screen, and this organization has fascinated researchers. So people will diagram the SCN sort of like this. My lab likes to call this diagram the chicken leg and egg diagram. It's meant to show you the neurons within the SCN organized into that shell and core regions. And the idea is that they communicate with each other to synchronize their daily rhythm. And different cell types may be able to talk to each other within the SCN and communicate out to control different behavioral and physiological rhythms. So this simple model has been evolving over the years. I'm just going to summarize for you something that you can look up, a really beautiful review by Jan Ray Silver. It tells you about how we've changed our thinking from thinking about the SCN as just containing a clock mechanism to thinking about how it contains a core and a shell, and that core and shell can communicate with the brain through signals such as diffusible signals or synaptic signals to drive daily rhythms behavior. That idea is going to evolve, and I'll show you data for this in a minute, where individual cells are capable of generating the 24-hour rhythms on their own, and that they must synchronize to each other, and that some cells may be more capable of sustaining rhythms than network of oscillators can have a wide variety of behaviors, including under extreme conditions in constant light. It can actually show anti-phase oscillations between the ventral and the dorsal SCN, and that may actually underlie behaviors such as splitting, which can be in some extraordinary cases, left-right splitting or dorsal-ventral splitting within the SCN to mediate 2 bouts of sleep per day instead of just 1. Ultimately, what we need to, as a field, understand is how is the SCN wired together and how does it wire into the brain and the rest of the body to control daily rhythms. So far, I've told you about the behaviors that we study and some of the markers for the SCN, but I haven't told you about how we study the SCN itself. So one approach is to reduce the problem to an SCN in a dish. And we can study the SCN in a dish in a wide variety of ways, recording, for example, electrical activity. And one technique that we use is a multi-electrode array like this one, where you have 60 wires on the bottom of the dish, and the tips of those wires are about the size of a neuron, and you can record the electrical activity or the action potentials of individual neurons. And if you count up those action potentials over time, you see daily rhythms and firing rates for as long as you care to record. Here's a recording for something like 15 days. I think the longest recording we've ever had in my lab was right around 30 days of daily rhythms and firing. And the only reason we stopped was because we got bored. And what's striking is you can resolve the activity of individual cells. And if you plate SCN cells on a dish in a way so that the cells are far away from each other and they can't really talk to each other very easily, you can actually see what David Welch reported in his seminal paper in 1995, where you can see daily rhythms in individual cells. Here a blue cell and another cell, a red cell, that have different periods in the same dish. This was really strongly suggestive that individual cells may be functioning as independent oscillators. We went on to do one additional experiment where he added tetrodotoxin, a blocker of voltage-gated sodium channels. So what happens is during that time, the cells cannot fire action potential. And then he washes off the blocker, and the cells resume their daily rhythms and firing. What's striking is it almost looks like the cell that went into the TTX came out of the TTX at the same phase that it had before, as if it was able to keep on ticking, even though there was no electrical activity or firing or communication between cells. So these data suggested that individual cells are capable of circadian rhythm, but it wasn't really clear which cells within the SCN could have daily rhythms in their activity. And if it was all of them, or maybe just a couple of grandmother cells that tell everybody else what to do. So what I'm going to do now is tell you a little bit about another technology that was first developed in circadian rhythms in plants and then applied to mammals. And this is the use of luciferase as a real-time recorder. So instead of recording electrical activity, which we think of as being sort of a hand on the clock, we're now going to use luciferase to record daily rhythms and gene expression. And we're going to focus in on genes that we think are essential for daily rhythms in cells. So you guys have probably heard about the transcription, translation, negative feedback loop, or the TTFL, which won the Nobel Prize in 2017. And in mammals, one of the essential genes that's been identified is the period 2 gene. Period 2 is an important gene in both rodents and in humans. Humans that contain a particular polymorphism in the HER2 gene wake up every day at about 2 o'clock in the morning, and then they go to sleep every night at about 7 o'clock at night. If you knock out HER2 in mice, you can actually end up with mice that have very long periods of rhythms that can turn into 8. We're interested in monitoring the expression of HER2 as a way to monitor the activity of individual cells within the SCN. So to do that, we take, for example, the motor of the HER2 gene, and and we hook it up to a reporter, in this case, firefly luciferase, an enzyme that's required that can mediate bioluminescence in, for example, fireflies. And it makes fireflies' bugs glow when it interacts with its substrate, luciferin. So luciferase is going to be, its activity is going to tell us when this gene is turned on and where it's turned on. We can take a slightly different approach, and we can take the full open reading frame of a gene and hook it up to the firefly luciferase. For example, we can now look at the expression of a purging protein fused to the firefly luciferase. Or you can do other approaches where you can just have a cell type specific promoter and get our reporter to be expressed in only some cells. For example, in this case, I'm showing you using a synapsin promoter driving the expression of GCAM, a protein that's derived from jellyfish to report how much calcium is in a cell. And all of these approaches have been used to show similar things that I'm going to show you now. My lab was interested in understanding which cells have circadian rhythms in the SCN, and we used that HER2 reporter, and we recorded from individual cells plated on cover slips for the SCN. And we recorded for a couple of days, and then the student in the lab, Alexis Webb, went in and she stacked all the cells in the dish, except one. She spared one cell, and that remaining cell was able to show daily rhythms in gene expression all by itself in the dish, demonstrating that, in fact, single cells truly are cell autonomous. And she repeated this experiment on many different cell types within the SCN, including the AVP cells, the VIP cells, and other cell types. And she found they were all capable of expressing sustained circadian rhythms in isolation. One thing was different. When these cells were reported all by themselves, when they weren't allowed to connect with each other, they were pretty sloppy. Their day-to-day period would be highly variable. In some days, they weren't even rhythmic at all. So we think that synchrony between the cells helps them become more precise and reliable, and that by themselves, SCN cells are sloppy circadian oscillators. So what I've told you so far is that the SCN is a multi-oscillator clock, and this is an illustration of the biological clock from a Hebrew, from an Israeli newspaper, where this says biological clock in the SCN. We know that the SCN is circadian in vivo and in vitro is required for daily rhythms in vivo, and is sufficient to rescue circadian period. It determines the timing of the behaviors and physiologies in our bodies. It's comprised of many different cell types, and now what we want to do is know how those cells synchronize to each other. So if you watch an SCN by itself in a dish, you'll see beautiful daily rhythms, for example, here in that HER2 gene expression that I told you about before. And what's extraordinary about these rhythms is there's sort of a wave of activity as we watch day to day. The rhythm sort of starts at the top of the SCN and then goes to the bottom of the SCN each day. And I want to take a minute to explain how we can measure synchrony. Because we're also interested in rhythms, we often plot a time series like this, where we see cells that appear to be giving daily rhythms and peaking at about the same time. Another way to plot those same data is a raster plot, where now each cell is a line, and you can see peaks and troughs in the expression of each cell. And you can see that all the cells are stacking up roughly in synchrony here for 1, 2, 3, 4, 5 days. Another way to quantify this synchrony is using what we call a Rayleigh plot. So a Rayleigh plot will take 1 day on a raster plot and present it as a circular day. And in that 24-hour day, we can plot the time at which each cell peaked, for example, in its firing range or its HER2 expression. And here we can see that the SCN cells stay very tightly synchronized for many days. But if we contrast that to the same reporter, or HER2 reporter, in fibroblasts, here data reported again from David Welch, we see that fibroblasts never really have synchrony. They start out in different phases on the first day, and by the cells within the SCN synchronize to each other. I'm going to tell you about one class of cells, the VIP cells. Those cells in the ventral SCN that express vasoactive intestinal polypeptide to show these cells are of particular interest to us because they are thought to be directly retinal recipient. They're the first line of response to light in the environment within the SCN. And what we can see is if we now use a real-time reporter of calcium in the VIP cells in an awake behaving animal, where we drop a light pipe into the brain, down into the SCN, we record the calcium activity during the end of the day and during the start of the day and throughout the day. We can see that during the day there's an increase in calcium activity in the VIP cells, and then that activity drops off at night. That persists in constant darkness here, and if we turn the lights on constantly, we see that day-night rhythm is abolished. Daytime light is where nighttime light is now driving activity in these VIP cells. And interestingly enough, if you have bright enough light, you can actually abolish rhythms in locomotion in rodents. And we think that may be mediated through this drive of the VIP cells. So are these VIP cells really playing an important role in synchronizing the SCN to the environment? One experiment we did in our lab was we actually silenced these cells so that they were unable to fire. And we found that in fact that blocks the ability of light to cause phase shifts in locomotor activity in neurons. We then turned to is activation of these VIP cells sufficient to drive and entrain daily rhythms in mice? And to do that, we took advantage of another technique where we can express a molecule called channel rhodopsin, which was stolen from bacteria in the salt flats of Israel, and we now use it in mammalian cells as a way to take control of the electrical activity of cells. So channel rhodopsin is a channel that is light sensitive, and when it's activated by light, it lets sodium flow into the cells and it depolarizes or excites the cells. So if we express channel rhodopsin only in the VIP cells of the SCN, and then we illuminate the VIP cells here where you see the blue bars every day for about 30 minutes, you can see that locomotor activity of the mouse synchronizes to that stimulation of the VIP cells. We played around with different patterns of stimulation and found that when these cells fire in patterns like this, bop, 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 they are capable of training the behavior of the animal very quickly. Other firing patterns, such as tonic firing, this pattern, bop, 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 can also entrain, but they're less effective. We see that these cells need to fire in small bursts to release their neuropeptide to help the animal train to the life cycle. So what about the role of VIP within the SCN? How does it work? Well, one thing we learned is that if we knock out VIP, the beautiful daily rhythms that you see in the SCN are still there in individual cells, but the cells no longer have synchronized rhythms to each other. They lose their circadian synchrony. So we think that VIP is required for synchronization to the light-dark cycle and for synchrony within the SCN amongst SCN cells. So we think that these VIP cells are sort of the first wave of response and coordination of daily rhythms within the SCN, and they coordinate with many circadian pacemakers. If we expose an animal to a shift in the light-dark cycle, something like a jet lag paradigm flight you from India to St. Louis, you can imagine that it takes some time to adjust. And recently work from Sang's lab by graduate students developed real-time reporting of VIP cell per gene expression and calcium expression trace as an animal was exposed to a shift in a light-dark cycle and then put in constant darkness. Here you can see the VIP cell activity gradually shifting over to the new light-dark cycle. So we can see that they are actually leading the response within the SCM to adjust to the new light-dark cycle. One other thing that I want to say about these VIP cells was discovered by Steve Brown, who I wanted to celebrate today. Steve was an extraordinary scientist. He was bold, he was brilliant, and he was also extraordinarily generous. Here he's shown with his wife, Patricia, at a conference last summer in Switzerland. And he hosted this meeting and then generously took everybody at the meeting up to the mountains with his giant bulldog and showed us how he loved the mountains. He was actually a remarkable mountaineer. He summited many of the world's highest peaks, including some peaks that were summited by only a handful of people. He was planning a trip this year to climb a mountain in Pakistan, I understand, taking a route that had not been taken before. So Steve asked many questions about the circadian system, and one of them was, which are the cells that mediate the daily siesta that we often see? And here I'm showing you local motor activity in a mouse, where the mouse is active for the beginning of the night, then it takes a break and it actually sleeps. He calls that the siesta. And then it shows another mouse bout of activity before it rests throughout the daytime. So Steve performed many experiments, and in a recent paper showed really beautifully that if you activate those VIP neurons in much the same way that I just showed you, we activated them on a daily basis, when you just activate them for a short time on one day, what you see is that you actually increase the propensity of the animal to go to sleep. And he argued that if the animal is relatively sleep deprived, these VIP cells can promote sleep and can drive that siesta after a long day of activity. So if you guys are beginning to feel a little bit sleepy after a long day, it could very well be that your VIP cells are contributing to driving you down to sleep. So as students, I'm going to give you this challenge. Consider this simple set of circles, dots, and lines as inputs, oscillators, and outputs. Are you now prepared to talk about the retinal input to the SCN and the different cell types of the SCN, to construct a basic model of how you think the SCN is entrained by light and can coordinate its own activity, and then drive and coordinate daily rhythms in things like the liver? I hope you guys have learned a little bit about the cell types in the retina that contribute to this entrainment. If you want to, we can discuss this later. So in the final minutes now, I'm just going to talk about future directions. I think one of the exciting directions for us now as a field is that we can lead the world in beginning to understand functional connectivity. So there's a tremendous amount of energy going on in neuroscience today, analyzing anatomical connections, mapping out where neurons make synapses in the brain. We think that this SCN circuit and its connectivity within and to the brain provides a huge opportunity to understand how daily behaviors and daily physiologies are coordinated through functional connectivity that may change as cycles like seasons as the days are getting longer. I'd like to compare where we are in the SCN with where folks were about 20 years ago in another neural circuit, a neural circuit that generates rhythms in chewing called a central pattern generator. They're very similar in many respects. The work by E. Marder and her colleagues has shown that this small population of neurons can generate very reliable rhythms through known connections. They've mapped out all the connectivity, and that those rhythms respond to sensory input just as the SCN responds to sensory input, and synapses. The SCN is 100% making it a very interesting circuit to study. And then these connections can be modified to produce novel behaviors depending upon the strength of those connections and their number. So I think that really does make the SCN an exciting place for mapping out on functional connectivity. In addition, I think we need to now start moving from the SCN to the rest of the brain and the body to understand how connections are, how we can get different outputs that are peaking at very different times of the day. So how do we get different phases of outputs from a population of cells that in mammals are, in rodents, are peaking during the day, regardless of whether the animal is diurnal or nocturnal. And finally, I think this connectivity should be studied as a function of things like age, sex, and disruptions such as exposure to, oh my gosh, I'm so sorry. Disruptions such as jet lag-like experiences. So beautiful work by Jen Evans and her team has shown that the SCM really does change its wiring so that the core and the shell can become very distantly separated in time, in terms of their time of peak per expression on very long days. So what are the connections that mediate and sustain this adaptation to seasonality? And so I'll stop there and offer that there are many other resources for you out there. I like this book called Superchiasmatic Nucleus as a place to start, written by these three gentlemen here. This book gives you a really nice history all the way up to about, I think it was 1982. In addition, there's a beautiful online video. If you Google HHMI, Joe Takahashi, SCM movie, you'll find a short 5-minute video that you can use in your presentation that illustrates many of the points that I made today. And with that, I will stop and be happy to take your questions.
Are there any questions in the audience?
Aakansha, can you communicate this question about the siesta? How SCN control the siesta? Is it the post-lunch siesta, or at what time the siesta is controlled by SCN?
Okay, so there's one question from the audience that how is that siesta window regulated? How is SCN actually controlling that break window, that siesta window? And at what time, for example, I know that you talked about rats, mice. So in the night they're active. So can we consider it as like a post-lunch break or what kind of break is that?
Yeah, that's a great question. So, siesta in humans would be during the day, siesta in mice would be during the night. The idea is that it's the cumulative integration of these VIP cells of some sort of homeostatic signal. So you guys heard in the last talks about how people think about the longer we keep ourselves awake, the sleepier we get. That's this hourglass mechanism that's measuring how long have you been awake. You guys have been theoretically awake for a really long time, so that homeostatic signal has been building up. And your VIP cells, we think, are measuring that signal. And when that signal is high and their circadian rhythm says it's the right time, they start to fire and they will promote sleep to help you recover a little bit of that lost sleep, and you can take a siesta. So the siesta is a normal biological process. It's something we should all obey after our naps are actually biological, not which is accumulating during our long periods of wake. And then that signals the siesta. Aakansha, you also mentioned after a meal. That's an interesting question about why do we fall asleep after a meal. I think that may be separate. I think that may be a different mechanism that's promoting sleep. I know less about that, but it's interesting to think about that the VIP cells may also be interested in signals that peak after lunch. That would be fun to check.
Sure, thank you. Any more questions? So there's another question that how SCN is connected to different brain regions related to activity behavior.
Beautiful. So it's clear that the SCN is required for daily rhythms and locomotions. What is unclear is the circuit that follows. The SCN can be transplanted inside of a dialysis network, inside of a tea bag, and it can still restore rhythms in locomotion. So this has suggested that the SCN may be able to reach the diffusible signal, and researchers have been working on this for about 10, 15 years to identify that diffusible signal. And I would say we do not know what that diffusible signal is. Several candidate signals have been suggested, including TGF-beta, but I don't think it's TGF-beta. That's a little bit controversial for me to say to you. Other things are also true. The SCN does make synapses to many other regions, mostly within the local hypothalamus. So it projects, for example, up to the subparaventricular zone and the paraventricular zone. And both of those locations seem to be very important for the SCN to communicate to drive daily rhythm. So if you lesion those areas, the daily rhythms in locomotion are disrupted to some extent. But it looks like there may be sort of multiple parallel conductive pathways that sort of protect and shape your daily pattern of activity. So different regions of the brain may be mediating the beginning of your activity, and other regions involved in the end of your daily activity. But the specifics are still being worked out. It's a really interesting area.
Any further questions? Well, I have one. How do you think VIP is associated with the morning and evening oscillators? So, is it also associated with somehow regulating the morning and evening oscillators separately, or are there altogether different peptides that are associated with it?
That's a really good question. So there's been some hints that under certain conditions, the VIP cells become the morning cells, and the AVP cells can become the evening cells. Those conditions are under extremely long days. We have pretty good evidence that the SCN can separate into a dorsal and a ventral oscillator component. However, it's not that simple, Aakansha, that we can say there's always a dorsal evening oscillator and always a morning ventral oscillator. I think that the connectivity and the identity of these cells is changing under different conditions. For example, in short days, it's not as simple as those two oscillators come together and become more difficult to separate. I think there's some mixing of the cell types and locations of cells and their contributions that we haven't yet figured out. And there are really interesting conditions where, for example, environmental cues might be environmental cues in addition to the light cycle may be contributing to how a cell associates with the morning or the evening oscillators. For example, right now you are eating during your sleep and you're sleeping and starving. drive anticipatory activity to go out and look for food. And those pacemakers may not even be in the SCN at all.
That would be interesting to look at. Thank you so much.
Thank you for the great question.
So Eric, it was really excellent talk. For you it's morning, for them it's evening. So they are in morning and different evening oscillator. And you would have all liked you to listen much more, but as the time goes by. And thank you so very much, and hopefully see you in less than 3 months' time in Washington, right? Yeah, so I'll start writing to those people. Thank you so very much again for spending time this morning, and as always, you were excellent. And my revision has been done at least for the next 6 months about the SCA. Thank you so much. Thank you.
Happy New Year. Bye-bye.
Bye. For today, so tomorrow we meet again at the same time here. Tomorrow I'll